Everybody, welcome back. Miss Calabrese here. So in this video, we're going to be talking about microbial metabolism. Right? So metabolism is just a sum total of all of the chemical reactions that are going on in a cell at any given time. So metabolism is all those chemical reactions that build new molecules, that break molecules down. It's how we get energy from our food. It's pretty much how we do everything inside of our cells. Right, so we can separate the concept of metabolism into two basic categories, and those would be the catabolic reactions and the anabolic reactions. So catabolic reactions are reactions that break molecules down. Anabolic reactions are reactions that build molecules up. So if we add together all the catabolic reactions and all the anabolic reactions together, then we get your full metabolism. Right. A lot of times when catabolic reactions occur, they release energy. So we take these large molecules, we break them down, and there was energy stored in the bonds that were holding those molecules together. So we release energy as catabolic reactions move forward. And so that means that a lot of times we couple those reactions with anabolic reactions because anabolic reactions need energy to go forward. So they can just use some of the energy that was released from a catabolic reaction to power them forward. So we kind of need both to happen at the same time. All right, so energy in general, we should talk about the different forms of energy that you use. Energy is just the capacity to do work, and for living things, that work is staying alive. So we can talk about energy from two different forms, either potential energy, which is a stored energy like the kind that we have in chemical bonds, or kinetic energy, which is the energy of movement, so actual motion. All right, in your cells and in microbial cells, the energy that's stored in atoms to fuel the function a lot of times is in the form of high energy electrons. So those electrons that orbit around the nucleus of an atom, those electrons can carry a lot of energy. All right. All the energy on Earth is going to come from the sun. We are, as humans, are not really good at harvesting that energy directly from the sun, but other critters are. So autotrophic organisms are ones that are able to feed themselves by converting solar energy into chemical energy. They basically can make food from sunlight. We are considered heterotrophs because we have to eat the things that we're making that energy. We can't make our own energy. So we can get the energy from plants by eating them, and the plants got their energy from the sun. Okay, so this free energy is the capability to do work. Sometimes that work involves building new molecules, right? So other terms that we should be familiar with here are exergonic reactions, which are reactions where energy is released, and endergonic reactions, which are reactions where we need to put in energy in order to get the reaction to go forward. So if we're looking at a chemical reaction and we can see reactants on the left side of the reaction and products on the right side. Exergonic reactions, the reactants have more energy because energy is released, and in endergonic reactions, the products have more energy because energy is required. Right. Other terminology we should be familiar with are the terms oxidation and reduction. So if we're talking about the movement of electrons, electrons, when we remove an electron from an atom, we consider that atom to have been oxidized. So oxidized molecules are ones where the electron has been removed. An electron has been removed. That's an oxidation reaction. If we receive an electron, so a molecule that takes on a new electron, we consider that to be reduced. So reduced molecules become more negative because they're taking on negatively charged electrons. Oxidized molecules become more positive because they are giving away those negatively charged electrons. And again, you can imagine if one molecule is losing an electron and one is gaining, that those are probably happening together. So oxidation and reduction occur at the same time, and we call that a redox reaction. All right, so let's talk about some of the ways that your body moves these electrons around. One of the common ways for some of the reactions we'll be talking about today is through electron carriers. These electron carriers like NAD, NADP, FAD, these are molecules they're organic molecules that were derived from B vitamins. So it's one of the reasons why B vitamins are critical to your metabolism, is because they act as these electron carriers to help move electrons around as we're undergoing important reactions inside your cell. So those forms are the oxidized forms, NAD plus, NADP plus, FAD. And then the reduced form is when we add an H on there. So NAD plus gets reduced to form NADH. All right, so let's talk about the major form of energy in our cells. A lot of this energy gets carried inside the molecule ATP. So ATP stands for adenosine triphosphate. And if we were to look at an ATP molecule, it basically looks like a nucleotide. So there's a 5 carbon sugar, a ribose, there's an adenine, and then there's phosphate group. But instead of just having one phosphate group, ATP has 3 phosphate groups. And those phosphate groups are negatively charged groups that kind of want to repel each other. So the fact that they're bonded together means that there's a lot of energy stored in those bonds holding those phosphate groups together. Right, so if you think of an ATP like this molecule right here, where we've got these 3 phosphate groups tacked on to the base of the molecule. There's a lot of energy that's stored in these bonds that are holding the phosphate groups on. So if we want to use some of this energy, if you want to think of ATP as like cash that you want to spend, there's potential energy in that $20 bill. We have to break it. We can't just if you can't spend your money and keep it too, right? So if we want to spend some of this energy, we have to break off one of these phosphate groups. Right, so if we break off one of those phosphate groups, now we're down to ADP, or adenosine diphosphate, because there's only 2 phosphates left. Right, so we were able to spend a little bit of that energy, but now we're left with something that's worth less. Right, so this ADP is not worth as much as an ATP in terms of energy. Right, so if we want to regenerate some energy, basically we got to go to work. Right, you can't just make money sitting around. So in order to turn this ADP back into ATP, we have to do some reactions where we're converting energy in order to be able to stick this inorganic phosphate back on the ADP in order to regenerate ATP. Okay, so ATP is your cell's currency, right? This is the molecule of energy that we use to power all the metabolic reactions in your cell. These metabolic reactions often occur in pathways, and that means that instead of looking at it just like as a simple chemical reaction where we've got reactants on the left and products on the right, in these metabolic pathways, we're going to have, say, maybe some reactants that yield some products, and maybe those products are going to be the reactants for the next step, and maybe those products will be the reactants for the next step. So we have several multi-step pathways here. That example is a linear pathway because we're just going in a straight line. We can also have branched pathways where we have one starting compound but then 2 endpoints. And then we could also have cyclical pathways where we've got a starting point, but we're continuously going in a cycle, and we'll have some molecules jump in and jump out, but we're just basically going around and around. Right? So these are the different type of metabolic pathways that are happening inside your cells. Okay. So we mentioned this concept of endergonic reactions, reactions that take energy to move forward. So if we were to graph the amount of energy needed, it would look a little something like this, where we've got the reactants over here that are at a very low energy state. And we've got the products over here that have a higher amount of energy than the reactants. So that means we had to add energy to move from the reactants to the products. That amount of energy necessary to get from point A to point B here is called the activation energy. So activation energy is the amount of energy required to push a reaction forward from reactants to products. Now, activation energy can be quite a large amount of energy. So in the green arrow here, we can see that's a high difference between the reactants and the top of this peak up here. So that's a lot of energy. Your cells have a way around this, though. So we can use catalysts to help speed up these reactions by reducing the activation energy. So a catalyst is anything that's going to reduce the amount of energy necessary for a reaction to go forward. In your body, you have biological catalysts, and those biological catalysts are called enzymes. What they basically do is reduce the activation energy. So now instead of having to go way up here, we only have to go here. So they're basically like metabolic coupons, right? They reduce the cost of getting from point A to point B, right? So they reduce our activation energy. Right, so here's another way to visualize that. So, and this one in particular, this one is going to be an exergonic reaction. So we have higher energy reactants and lower energy products, but there's still a hump of activation energy that we need to get over to get down to those products. So again, this is where enzymes would come into play, acting as these biological catalysts that are going to reduce the amount of activation energy so that this reaction can happen more quickly. A lot of the reactions in your body would happen regardless of having an enzyme present, but the enzyme will speed it up because without that, the increase in speed, those reactions would be happening so slowly as to be incompatible with life. All right, so here's a way that we can visualize enzymes. So enzymes are proteins. That means they're really large, kind of complex molecules that have very specific shapes to them. And that shape is going to be critical for their function, right? So structure equals function here. If the shape of our enzyme is wrong, it won't be able to do its job. So here the enzyme is represented as this kind of gray blob here that's got this really interesting shape on one end. We call that the active site. That active site is where a particular substrate is going to fit in. So for every reaction that we need to get done, we're going to have a specific enzyme that's capable of doing that reaction, and usually only one enzyme for one reaction. And that's because of the interaction between the enzyme and the substrate. So we can see here that the substrate has a very specific shape, and it happens to mirror the active site of this enzyme. Right, so this enzyme will only be able to act on that particular substrate and not act on any other substrates. So if we imagine here that this enzyme is the enzyme catalase, and let's pretend that this substrate up here is the molecule hydrogen peroxide. So hydrogen peroxide is a molecule that's produced in your cells as kind of a byproduct of some other reactions. It's toxic, and so we usually want to get rid of it fairly quickly. So when we have this hydrogen peroxide produced, we need this enzyme catalase to help us break it down. Um, so the hydrogen peroxide fits nicely into the active site of the catalase molecule. And then catalase is going to do its job by rearranging the atoms in this molecule and converting one hydrogen peroxide molecule into one water molecule and one molecule of oxygen gas. Right, so it's broken down the H2O2 into H2O and O2. Right, and this is a reaction that will only happen with catalase. You won't be able to use any other enzyme to break down hydrogen peroxide, and you also won't be able to fit any other substrate into the enzyme catalase. So the two work together, and they work out almost like a lock and key formation, as there's not going to be any other key that fits into this lock, and there's no other lock that's going to work with that particular key, right? So, and that's how enzymes basically help us carry reactions forward. Also, they don't get used up in a reaction. So a single molecule of catalase will be able to catalyze maybe not millions, but thousands of reactions per second. It will just keep doing this over and over and over again without being used up or without getting worn out. Right, here's another way to visualize it. So again, enzyme, really large protein, complicated shape, but it always has an active site, and that active site is going to be specific to a particular substrate. And so once that substrate kind of nestles itself into the active site, then the enzyme can do its job, rearrange some of the atoms in that molecule and cause whatever's happening here, which seems to be a decomposition reaction. Right? Oftentimes enzymes don't work totally on their own. So sometimes they need help from other molecules in order to be able to accomplish these reactions and speed them up. When they need helper molecules, we refer to those helper molecules as cofactors. So cofactors can take one of two forms. They can be inorganic molecules like metal ions, like magnesium, zinc, copper, or they can be organic cofactors that are made from vitamins. And those would be, if they're organic, we refer to them as coenzymes. Right? So cofactors are inorganic, coenzymes are organic molecules that are going to help process this chemical reaction. And basically, they're helping to rearrange some of the electrons, hanging on to electrons as we're moving other molecules around. So that's where these electron carriers come into place. So these are cofactors that are going to help progress reactions forward. All right. Enzymes can also be kind of temperamental. So because they're proteins, and proteins have very specific structure that results in very specific function, if we change their environment, sometimes that can result in a change in the shape of the protein, which will result in a change in the function of the protein. So things like temperature, pH, pressure, all of these things can have an effect on the function of a particular enzyme. So in this graph down here, we can see at low temperatures, this particular enzyme doesn't function that well. If the temperature goes up, it's going to function a little bit better, and all the way up until we hit our optimum, right? So whatever temperature this is, that's going to be the optimum temperature for this particular enzyme. And if this is one of the enzymes in your body, then it's likely going to be working its best right around 37 degrees Celsius. And if we go too far above that, so if we go to a temperature way out here, we can see that the enzyme activity really drops off. It basically crashes. And that's because at temperatures above that optimum, you're essentially melting the protein. The bonds that were holding it in that very particular shape have denatured. So we consider that a denatured protein because it's not the same shape as it was, which means it can't perform the same function of catalyzing that reaction. Similar thing is true for pH. So most enzymes have a very particular pH that they like to function well in. This one's a little bit more variable inside your body. So you've got areas in your body that function really well at a pH of maybe just above 7. And you've also got some areas in your body that function really well at very low pHs, like in the contents of your stomach, for example. Enzymes present there really like to function at a pH of around 2 or 3 versus enzymes that are in other places in your body, which might like to be between 7 and 8. Okay, so say that we want to change the effect of an enzyme. If we want to slow it down or inhibit its function, there are a few ways we can do that. One way is through competitive inhibition, and competitive inhibition basically involves the use of a molecular mimic or something that a molecule that looks like the substrate but is not the substrate. So here, in this example here, we've got an enzyme down here with an active site. That active site will fit perfectly with this particular substrate, but also we might be able to create a mimic that can fit in there instead. And if that mimic fits in there instead, he's going to block the actual substrate from going inside that active site, which will block the reaction from going forward. So this concept of competitive inhibition relates to us in microbiology because this is one of the things that we use to create a whole class of antibiotics called sulfa drugs. So sulfa drugs are ones that prevent folic acid synthesis in bacteria, and they do it by creating this molecular mimic that will stop the active site of that particular enzyme. All right, so another kind of inhibition that we can do is called non-competitive inhibition. Non-competitive inhibition, well, one particular type of that is called allosteric inhibition, which makes more sense to look at a picture. So I'm going to skip ahead to this slide here so we can see what this allosteric inhibition looks like. So here's our enzyme, right? And then here's the active site of the enzyme that's going to fit perfectly with this particular substrate. And in competitive inhibition, we would have a mimic come in here and block. But in allosteric inhibition, we actually bring an allosteric inhibitor into the other side of the molecule, right, to this allosteric site. And that, when this allosteric inhibitor bonds to that allosteric site, it's going to change the shape of the active site temporarily, right? So it changes the shape of the active site temporarily, which means the substrate can no longer fit into it, and that's going to stop production of whatever this pathway was doing. So a lot of times, your cells use this as kind of a feedback, meaning that if we're looking at this metabolic pathway down here, we've got some starting compound, we've got some endpoint, but that endpoint acts as an allosteric inhibitor, which means that endpoint product can go all the way back here and inhibit the function of enzyme A, right? So stop the pathway going forward because we've got too much product. It's basically a negative feedback loop that's going to shut off production until we've used up enough of that product that we need to start making more of it. Right? So that's how non-competitive inhibition works, specifically allosteric. All right, so let's move into the process of respiration. All right, so we've talked a lot about ATP and how ATP is going to be an important energy molecule for all the metabolic reactions that are going on inside your cell. We make ATP in a couple different ways, but the main way is through oxidative phosphorylation. And I know that's a big word, but it just means phosphorylation means to add a phosphate group, right? So we're adding a phosphate group. Oxidative means with the use of oxygen, right? So we're using oxygen to add a phosphate group to an ADP to convert that ADP to ATP, right? So that's all. It's a lot of fancy terminology, but we're just creating an ATP molecule from an adenosine diphosphate by using oxygen. All right, so here's what that looks like. So this chemical equation down here, you're going to need to know this one. It's not balanced. So for those of you that are experienced in chemistry, I left it unbalanced here just to keep it simple. So right here, this first molecule that you're looking at, this is glucose. Right, so consider this food. For everything that you eat, we're gonna say that that is food. That's glucose, and that's going to be the starting point for our production of ATP. So you need to eat food to get the energy from the food. All right, so the food is going to be represented as a glucose molecule here, C6H12O6. We also need oxygen. So this is the entire reason that you breathe in oxygen. There's nothing else your body uses oxygen for, except to help break this glucose down and turn it into ATP. So these are our reactants. So this is what goes into this process of aerobic respiration. What comes out the other side then is CO2, right? Obviously you exhale CO2, that's a waste product, you don't need that. We produce a little bit of metabolic water here, which can be recycled and used inside your cells. And then the big endpoint that we're looking for is ATP, right? So we're getting that ATP from the energy that was stored in the bonds of this glucose molecule. Right, so we're turning glucose into ATP by breaking the bonds in there and converting the energy in those bonds into the energy that's holding the ATP together. All right, so we're going to break this down into 4 major steps. We're super oversimplify the process and say there's basically 4 steps here. The first step is called glycolysis. Right, glycolysis is simply the breakdown of a glucose molecule from one 6-carbon sugar, right? We can see there's 6 carbons here, one 6-carbon sugar into 2 3-carbon molecules of pyruvate, right? So one glucose becomes 2 pyruvates. When we do that, when we break that glucose down into 2 pyruvates, we're going to release a little bit of ATP. Right, so we're already getting some energy out of breaking this molecule down because there was energy in the bonds holding it together. So we're going to get a little bit of ATP. In addition to that ATP, we're also going to reduce some NAD. Right, so that means we're going to convert it from NAD to NADH. Right, so we're loading up an electron carrier here. We're giving it a high energy electron that we received from breaking apart that glucose. Right, so this process of glycolysis that's going to yield 2 ATP and also NADH is happening in the cytoplasm of all cells. So cells are doing this glycolysis process in the cytoplasm. The next steps of this process and what makes it aerobic respiration is if we have oxygen present and we can make this aerobic, then we're going to move into the mitochondria. Right, so the mitochondria, remember, is the powerhouse of your cell, and we consider it that because it's capable of carrying forward the rest of these reactions here. So next step is preparatory reactions, In the prep reactions, we're going to convert those pyruvates that we made in glycolysis into acetyl-CoA, or acetyl coenzyme A. That acetyl coenzyme A is going to enter into the next step, which is the Krebs cycle. In the Krebs cycle, this is going to be one of those cyclical pathways where basically we're going to load up some more electron carriers. So we're going to take our FAD, make it into FADH2. We're going to take our NAD, make it into NADH, and we're also going to get a little ATP out of this reaction as well. So again, this is happening in the mitochondria. So we're loading up more electron carriers. We're getting a little bit more ATP out of further breaking down what was left of that glucose molecule. And now we get to the last step of cellular respiration. This is kind of the big payoff. So this is the electron transport chain. So electron transport chain is when we're going to take all of these reduced electron carriers. So NADH, FADH, this one from up here. We're going to take all of these electron carriers and harvest the energy from them, right? So they're carrying all these high energy electrons. We're going to now take those high energy electrons and use that energy to convert ADP to ATP. So we're just going to use it to add on an inorganic phosphate molecule to an ADP in order to yield more ATP. And we can get up to around 34 ATPs from this process of the electron transport chain. Right? So it's a very efficient process. We can only do it if we have oxygen. If we don't have oxygen, then none of these mitochondrial steps happen. Right? We need oxygen in order for the mitochondria to be able to do its work efficiently. Okay, here is what glycolysis looks like. Don't just, you know what, just ignore it. Focus on what goes in, which is a glucose molecule, and then what comes out, which is a pyruvate molecule. So 1 6-carbon molecule goes in, 2 3-carbon molecules come out, and that's basically it. Other outputs that we see here is that here's an ATP, so we get an ATP out over here. Here's an ATP, so we get an ATP out over here. So there's 2 ATPs that we net out of this process. And then we also get our reduced electron carrier, which is the NADH. So that's what we're getting out of glycolysis. NADH, 2 ATPs, and 2 pyruvates. Here's a look at the Krebs cycle. So Krebs cycle is one of those cyclic metabolic pathways. So in here we can see that the acetyl-CoA is what's entering into the Krebs cycle here. And then I've also highlighted the inputs and outputs in green and red here. So, so the inputs are labeled in green. Acetyl-CoA goes in. An NAD goes in here, another NAD here, and FAD. So we've got these oxidized carriers. We're going to reduce all those carriers. So we're going to take the energy that was left in this acetyl-CoA and help load up these electron carriers. So we're going to create some NADH, more NADH. We're going to create some FADH2, more NADH. So we're creating some loaded electron carriers. We're also going to convert a little bit of ADP to ATP, so we do get a little ATP out of the Krebs cycle, but for the most part, what we're doing is loading up those electron carriers so that we can have these really high energy electrons ready to yield more energy in the next step of this process. Okay, all right, so yeah, this is basically saying that we're just loading up electron carriers that we've been doing the whole time. So we loaded up some in glycolysis, we loaded up some in the transition step or preparatory reactions, we loaded up more in the Krebs cycle. Now we've got a whole bunch of high energy electrons that are in these electron carriers, and we're going to take them to the last step, which is the electron transport chain. The electron transport chain, you can think of it as basically just unloading all of those high energy electrons. We loaded them all up. Now let's let them free. As we let them free, let's look at the next picture. There we go. So as we let them free, they're going to go through, help basically a turnstile, and help power this molecule of ATP synthase, this enzyme, to convert ADP into ATP. right? We're just using those high energy electrons to help power this reaction that's going to convert ADP to ATP. So at the end of all this, we're going to end up with a whole bunch of ATP that we manufactured basically from a single molecule of glucose, right? All of the energy stored in that glucose molecule gets broken down over a series of steps, and that's how we get all of this ATP at the end of the electron transport chain. And again, this is happening in the membrane of the mitochondria. Okay, what if we don't have oxygen? So in that electron transport pathway, I may not have pointed it out on that slide, but the final electron acceptor in that process was oxygen. And so we end up with some water. So that's where we get that water as an output of the cellular respiration equation. If we don't have oxygen, though, we can't go into the mitochondria and we can't do any of those fancy reactions. What will happen instead is fermentation. So fermentation is where we have to stop after glycolysis, and we're not going to be able to proceed because we don't have the oxygen necessary. And remember that oxygen was important to make the process efficient. So fermentation, we still get a little bit of ATP, so we'll still get a couple ATPs out of this process, but it's not nearly as efficient as the 34 to 38 ATPs we would get if we continue to go through the aerobic pathway. All right, so a couple different types of fermentation. The first one here is lactic acid fermentation, and this is the one that you do, mammals do. So a pyruvate, or all vertebrates, I believe, do this one. So pyruvate is that the result of glycolysis. That pyruvate, instead of getting converted in the preparatory reactions to acetyl-CoA, in the fermentation reaction, is going to get converted to lactate or lactic acid. And that lactic acid can build up in your cells and cause kind of like a burning sensation. This is what happens if your cells run out of oxygen. Right? If they don't have enough oxygen for their mitochondria to function properly, they're going to still need to make ATP, and the best way they can do it is through lactic acid fermentation, which is really something only your skeletal muscles can do. And they only do it when they're really out of energy. So if you're working out, if you're running a race, if you're doing something like that, then you could possibly burn through all of your oxygen and be forced to go into lactic acid fermentation. And that burning sensation that you feel in your muscles when you're running past your oxygen threshold is the buildup of lactic acid. Other critters, a lot of microbes, use ethanol fermentation or alcoholic fermentation. So they're going to take that pyruvate that they got from glycolysis and through a series of pathways turn it into ethanol or just alcohol, right? So this is useful for us because basically we just need to take oxygen away from yeast and they'll give us alcohol. And so that's how we manufacture alcohol. All right, so a few questions here. The process of breaking down molecules, the process of building up molecules, which one is a type of metabolism, and which one releases energy? All right, so here's your answer. Catabolism is breaking down molecules. Anabolism is building up molecules. Both are type of metabolism, so we need both of them in your metabolic process. And then which one releases energy? That would be catabolic reactions. Right, a few more questions here. What does ATP stand for? What is it? Which molecule, AMP, ADP, or ATP, stores more energy? And then what is the term called when a phosphate group is added to ADP? So here's your answers. ATP stands for adenosine triphosphate. It is a molecule of energy storage, a major currency of your cell. Of those three molecules, adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate, ATP, the triphosphate one, stores the most energy. It's got more of those phosphate bonds. And then what is that called when we add a phosphate group? It's called phosphorylation. All right, last few questions here. So what is a molecule called if it loses electrons? What is it called if it receives an electron? And what is the reduced form of NAD and FAD? All right, so if a molecule loses electrons, it is oxidized. If it gains electrons, it is reduced. And the reduced forms of the NAD and FAD are NADH and FADH. All right, so I hope that was helpful. Let me know if you have any questions.